So, to start off with a little bit of background, I know that many of us here today study a particular disease, and so the disease that I'm interested in is cancer. And what kept me in the lab for pretty late hours during the start of my degree were cancer statistics. And so just to share those statistics with you, cancer is actually the #1 cause of death in Canada. 1 in 2 Canadians will develop cancer, and unfortunately, 1 in 4 Canadians will succumb to the disease. Now, while we should, of course, strive to improve these statistics for the particular cancer that I was interested in, I felt that this was actually quite a narrow scope, and it really only looked at the treatment portion. Now, the cancer journey is, of course, much more complicated than that. And while we can't cover everything, I wanted to not only look at the treatment, but I also wanted to consider what their quality of life was while they move from their treatment to their next steps. And so just to start off with a little bit of a summary of what we'll actually be covering today, I'll actually be giving you a bit of background for head and neck cancer, which is our cancer of interest. I'll tell you how I feel or how I think nanoparticles will be integrated into their treatment. Then I'll tell you how we actually synthesize these nanoparticles, how we characterize them, how we evaluated with them within our system, and of course, the many next steps that we still have before we make it to the clinic. So just for a bit of background for head and neck cancer, about 60% of patients come into the clinic at a late stage of the disease. And when this happens, the gold standard for their treatment is actually a combination of both high dose cisplatin, so chemotherapy, as well as high dose radiation. And I take it that you probably won't be surprised when I say that 40% of these patients show very significant toxicities because you're combining already two toxic therapies. And so for us to solve this problem or to remedy this problem, we actually propose to use a nanoparticle system. And in order to do that, we're hoping to sequester that cisplatin, so that chemotherapy, within a nanoparticle so that while it's circulating systemically, it won't end up in organs that it shouldn't be in and cause those toxicities. And then once it's actually at the tumor, we're going to use that radiation that's already incorporated into the therapy to trigger the release of that drug. And thank you to Talia for talking about the combination of both gold nanoparticles as well as radiation, because we're also using this in our system and loading those gold nanoparticles within our carrier protein nanoparticle that will be carrying the drug. So again, just to reiterate what Talia had said, that those gold nanoparticles will help increase the local dose of radiation and will actually help cause that triggered release. And so why are we doing this? Why did we choose the components that we did? So we decided to go with a protein carrier to load both the drugs and the gold nanoparticles in there. And we chose a protein called Zein, and it actually comprises about 60 to 70% of the endosperm of corn. So it's extremely abundant. It's actually quite easy to extract, and it's already approved in Canada within the clinic for purposes used in, say, lymphangiomas. Now, while that's of course great, what I think is most interesting is actually the properties of the protein themselves. So most animal proteins are overall hydrophilic proteins. However, the Zein is actually hydrophobic, and it's comprised of these 9 alpha helices, and it forms this really interesting barrel shape that you can utilize in order to load chemotherapies. And this has been shown over and over again within the literature. So we saw this as a very interesting system to create our hybrid nanoparticles. So our first question, because we're more of a liposome lab, but we decided to use these proteins, was how are we actually going to synthesize this? And of course, we scoured the literature and we found that the most commonly used method was considered a one-pot synthesis. So you essentially have your beaker, you have your aqueous phase, which is water, and then you just pour everything into that same beaker, you stir it, and you hope you get the same thing each time. So of course, being very skeptical, this was, as you can see, not the case. So in the top left, I took my student at the time aside and I said, okay, let's do this experiment side by side and see what we get. So you can see in trial 1 and trial 2, we actually get very different results in terms of the size, as well as the dispersity or the differences within our own samples as well. And so the rationale for why it's so different and why both my student and I got such different results is because really depending on where you pour your sample within that beaker, it's going to be essentially introduced to a different environment, such as different shearing forces. So this makes it very non-reproducible and not ideal, of course, for a clinical formulation. So we decided to go with microfluidics instead. And microfluidics is similar to a computer chip, except instead of having electricity run across the chip, we actually have the two fluid components combine in very consistent ways. And you can see that this consistency is translated across to the size of the nanoparticles. As you can see over those 6 trial numbers, it stays the exact same size no matter when they were performed or what batch we use. So we were extremely thrilled to see the consistency of the system. And we were also very excited that this system was very tunable. So what I mean by that is at the bottom right, you can see the standard characteristics. And so you could easily modulate or change these standard characteristics to get very predictable changes in the size of your nanoparticles. So we wanted to continue with the synthesis using this microfluidics method due to the reproducibility as well as the tunability of the system. And so once we got down the synthesis method, we of course had to characterize our hybrid. So we used electron microscopy in order to do this. And you can see in A and B, which is our empty carrier, there's no gold. However, in D, E, and F, you can see the gold nanoparticles are together with the Zein nanoparticle. Now, while that's all well and good, that doesn't necessarily mean that these particles are associated. So after cleaning up our sample, we decided to characterize it further using cryo-TEM. And so there were 2 different scenarios that could really happen. So we took our sample and we tilted the stage, the cryo stage, and imaged the same particle at 3 different angles. And when this happened, if they were associated in scenario 1, for example, we would see the dark particles, which are the gold, stay associated with the Zein particles no matter what angle was at. However, in scenario 2, if they were not associated, once we actually tilted the stage, you would slowly start to see that these 2 particles would disassociate from each other. And so you can see by the orange arrows, which is scenario 1, where they would stay together, the black gold nanoparticles stay with that dark gray Zein nanoparticles no matter the angle that you tilt it at. However, in scenario 2, the red arrows, I had a little bit of fun and I made some bubbles using the electron beam, and then I tilted the system, and you can see that those gold nanoparticles start to drift away from that bubble because they're not associated. So we use this in order to actually characterize the association of our 2 different nanoparticles. So now that we actually have a hybrid, the next question was, can we actually use this as a triggered release system? And so like Talia had explained in her presentation quite well, was that the gold will amplify the interaction of radiation with surrounding material. And so when those X-rays interact with surrounding material, it'll cause something called reactive oxygen species. And as you would expect from the name, these are of course extremely reactive. So if we have these reactive oxygen species present, we expect them to interact with our protein. And if they reacted with our protein, we had expected 3 things to happen. One was the aggregation of our protein. Next was the fragmentation of our protein. And then lastly was the chemical modification of our protein. And we actually saw all 3 of these occurring at once. And so while we are excited, of course, by these results, we have many next steps that we need to take before going to the clinic. So the first is really evaluating the system as a triggered release system. And we used irinotecan just as a model, and we saw that only the ones containing gold acted as a triggered release system. So this was very promising. Next, we wanted to look at the toxicity of the vehicle itself, so just the protein and the gold by itself. And we saw that it was very non-toxic to normal as well as phagocytic cells. However, of course, our next steps would be to actually look at it with a drug loaded and show that it's efficacious within the cell system. Next, we of course want to check in vivo safety of our protein nanoparticle. And so we injected it and we monitored the mice over 14 days, and we saw that it was non-toxic and it didn't accumulate in any organs that we would consider dangerous, such as the heart or the lungs. So this was also very promising. And so lastly, one thing we need to evaluate is the immunogenicity as well as the pharmacokinetics of our system. So the key takeaways really are that hybrid nanoparticles exhibit changes when irradiated, and that these changes may be capitalized for use in triggered release. We, of course, still need to use this system with cisplatinum, have some efficacy studies at the cellular level, and then once we have that, we'll be able to move on to head and neck cancer mouse model. So thank you so much, and I look forward to your questions.
Great, thank you very much, Courtney. So I'm pleased to open the floor to questions from the audience for the last time today. Please either submit a written question or raise your hand. So I do not see any questions or hands at the moment, so that gives me a chance to ask a question. Could you speak to why gold nanoparticles were selected to enhance the trigger?
Yeah, no, definitely. So the gold nanoparticles, kind of what Talia was saying, was that because they're a relatively dense material, they'll actually be able to deposit a higher dose of radiation locally. And by having that higher localized dose of reactive oxygen species, we'll have more of those reactive oxygen species to then react with our protein. And then we'll get those 3 downstream side effects of aggregation, fragmentation, and modification. So yeah, that's how we hope to use it.
Great, thank you. And then could you speak to why irinotecan, I'm going to mispronounce that, was used for the drug delivery study? So I think you mentioned that on the next step slide.
Yeah, definitely. So we chose irinotecan originally just as a model or proof of principle, because if we had incorporated cisplatinum first, platinum is also a heavy element. So we thought that that might confound some of the results that we would get if we used a drug that was also a heavy element like gold.
Okay.